First of all, as you said, I'm professor of biochemistry, and I'm chairman of the Center of Excellence for the Harm Reduction at the University of Catania. And together with the Professor Caruso, I am a principal investigator on one of the 8 projects funded in our Center of Excellence that is called the Replica Project, that is seeking to replicate some of the most important work performed by the tobacco industry regarding the harm reduction strategies. So why do we care about replication? So first of all, we have to understand, and that I just learned from myself and with this beautiful experience, understand that replication is not reproduction. So you cannot get exactly the same results as other people are getting in lab. So if you want to measure after exposure with cigarette or with e-cigarette interleukin 6, and for just say they measure 16 picogram per ml, you can forget about you get 16.2 or 16.1. So what you want to do is just replication. So you want to demonstrate that the basic hypothesis is correct. So that harm reduction reduce inflammation as measured by interleukin 6. Doesn't matter if you get exactly the same amount of picogram per ml. It's really hard. There is so much variables that you cannot take into due account when you set an experiment. Then, of course, what we have to replicate, and I think that Professor Farsalino said at the beginning of his talk, we have to replicate only studies that are relevant. So what we decided to do with the replica project, I don't know if probably some of you had the chance to listen to Professor Caruso talk of yesterday. Of course, we want to replicate studies that have a profound impact on harm reduction strategies. So that's why we choose some of studies rather than others. And then, of course, we want to replicate in a way that we can generalize the original study. So we don't know, of course, we use most of the methods that was used in the study which we are replicating, but we always add additional methods in order to further demonstrate the hypothesis and to convince more about the and make the results more strong. Of course, replication is very important, and the problem with replication, and that is the most difficult part of the of replicating study, is to harmonize the methods, because of course what we do at the COIR is that we replicate in order to make sure about the conclusion. We replicate our studies in a ring trial. So we have 5 different labs all over the world, in particular Italy. We are the coordinating group here at the COIR. Then we have the University of Sultan in Oman, Muscat. We have Serbia, Greece with the Professor Konstantinos. We have Kazan, University of Kazan in Russia, Indonesia, and we have the Sbarro Institute at the Temple University. So the problem when you want to make a strong conclusion, you have to harmonize the protocols. That it looks simple, but even when you have a very simple technique in the lab, which may be a neutral red uptake, you can just do a simple ELISA test. Then when you want to replicate in 5 different labs, then it becomes a problem. So you want to use the same passage of cells, just making samples. You want always to use the same lot number of the serum that you want to use to culture your cells. You want to use the same kit, the same lot number, the same plastic, the same disposable, of course, all the protocols, same wavelength when using the spectrophotometer lectures. So you have to harmonize, and there is a very nice, even if not very recent work published in clinical chemistry that proficiency test most of the hospitals and laboratory has failed because the harmonization was not performed conveniently. And also in science, as I was telling at the beginning of my talk, that is a problem, the replication. There is a very interesting paper came out in May 2020 on Science in the Wire, that the papers that scientists could not replicate it were more cited, meaning that we have a problem with I don't want to say that people, they publish fake results, but no science is a perfect science. So even if you can make mistakes in your protocols, of course, most of the study, they are one center study. I'm talking about basic science. I'm not talking about clinical trials. Most of them are multicentric. So you are sure about your results you just published, but if nobody is replicating them or is able to replicate them, then there is no strong conclusion that science, and we cannot assume that as a correct data. So we are starting from this principle that no science is a perfect science, and if some study has to impact profoundly on public health, such as harm reduction, then we have the commitment as a medical doctor, as a scientist, to replicate that studies, to offer to our patients the best science possible and the right information regarding their personal choice of switching to a normal cigarette, to an electronic cigarette, or to harm reduction strategies.